I would now like to welcome to Centre Box Dr. Wendy Williams. Dr. Williams is from UQ. Her expertise is in the area of Biocrust in the Rangelands and Savannah. She's currently working on a project with MLA and about increasing nitrogen in the rangelands. She's working with the DPI in the Northern Territory and the Department of Agriculture and Fisheries in Queensland. So please welcome Dr. Wendy Williams for this next session.
Hello everyone, and welcome to learning about biocrusts today and what biocrust hotspots are and how we can include them in grazing management and biodiversity in Northern Australia. So first of all, I want to start off by talking about crusts versus crusts, because they're different crusts and people understand them differently. So you talk about a crust or a biocrust or a physical crust or a chemical crust. So in this little photo here, you can actually see the difference between three types of crust, because it's taken from a salt lake in South Australia. And you've got at the bottom here a crust which is photosynthesizing cyanobacteria, and it's living underneath a salt crust which has formed a physical crust. So these crusts can be confused in the field, but today we're talking about biological crusts or biological soil crusts, or biocrusts for short. So biocrusts are a living, breathing microbial community. They form a matrix, first of all, with cyanobacteria, bacteria and algae, and those microorganisms, the cyanobacteria, which you can see as a blackish cover on the surface there, they actually facilitate colonisation by other crust organisms, and it depends on where you live as to how many other of these crust organisms live there. So there's lichens, liverworts, mosses and microfungi. And as we inferred in that first slide, is that they actually live on the soil surface and between grass plants. They also capture and retain resources. They capture soil, they capture water, they capture seeds, organic matter, dust, minerals, and they enhance fertility through biological nitrogen fixation. To form, it's really good to actually understand, looking under the microscope, as to what these look like on the soil surface. They're in what we call the critical zone because they're right between the atmosphere, the soil surface, and what's underneath the soil surface. So you can see here in these micrographs and scanning electron microscopes, you get what they call EPS here, which is sticky stuff, and I'm sure some of you know what that means in the wet season, and we'll look at that in a minute. And then you get the little filaments here of cyanobacteria, which you can see the bacteria's part of the soil crust is too hard to see in this magnification, but you can also see the surface here, a cross-section, and how these organisms and filaments penetrate into the surface and how they bind the soil together. So in these areas, dry season biocrusts and wet season dry biocrusts appear quite differently. So the dry season biocrusts we often refer to as the crunch under your feet. So you walk through the paddocks and you can feel the soil crunching under your feet, and quite often this is the cyanobacterial soil crust which is on the ground, and you can see in the images here, they shrink and crack and curl during the dry season, and they create homes for ants and other little microfauna, and they also have little spots where seeds can penetrate into them. Whereas in the wet season, you get the slimy slippery time, or what I call the slimy slippery time, because you can fall over on them pretty easily. And the EPS, or exopolysaccharide, which is the slimy covering, holds that crust together. It starts green and then it goes into a black colour, and it spreads quite quickly. You can sort of go there one day and there's nothing, and then you go there the next day and there's quite a lot. So cyanobacteria are the ones that produce most of this EPS, or the exopolysaccharides, which is the slimy coating. And one of the unique properties of it is in Northern Australia, When it's in the dry season, it becomes hydrophobic, meaning it repels water, and in the wet season, it becomes hydrophilic, meaning that it absorbs water. And this is a special characteristic to preserve it against unseasonal rains. And it protects the cells, but it also acts as a dispersal mechanism, and it stores excess sugars. So currently, we're involved in research with MLA, Northern Territory Government, Queensland Government, University of Queensland, and the research program is called Boosting Natural Regeneration of the Nitrogen Capital in Grazing Lands. So the reason why we're looking at this as the nitrogen capital is because many of these cyanobacteria and bacteria that live in the biocrusts fix nitrogen, fix carbon, they photosynthesize, so it's important. So one of our research field sites is at Kidman Springs, and many of you have already heard about Kidman Springs from Robin Cowley just in the previous session. But in this experiment, we're looking at how fire impacts biocrusts and how do they recover after fire. So we can see in this area when there's been a fire through that it burns through on the soil. So we're actually interested in what happens after fire. To do this, we need to understand a little bit about landscape function and where the biocrusts live. So the biocrusts live in under the grasses, which are the patches, and in between the grasses, which are the interpatches. And they occupy the spaces on the ground. So if you have no biocrusts there, you can see what happens with the resources and the nutrients and the topsoil. It just erodes away. And if you have a heavy rainfall, as in this photo, then that can just wash away. But the biocrusts act as a protection to the surface because, as we explained earlier, they've got those sticky polysaccharides and that they actually help to bind the soil together. So our early results have shown that biocrusts are well adapted to fire. They recover after fire. They're at Kidman Springs. They're a prominent part of the landscape, and this fire experiment's been going for 29 years. So if they didn't recover after fire, they wouldn't be there after 29 years of fire regimes. So long-term fire cycles do not destroy biocrusts, but in our work we've noted that straight after fire they were depleted a little bit by the fire. So that did make a difference to the communities, however, they were able to recover. So we did a lab experiment and it showed that the cooler fires, so the early season fires, had higher carbon and nitrogen levels in the biocrust post-fire. We've also been looking at the Biocrust DNA. We've said, well, we really want to know who's there, what are they doing, what's their role in the system. So in this, we've taken samples from inside an exclosure where there's no cattle and outside the exclosure where there are cattle grazing. So that way we can have a little bit of an idea of who's actually there. So this Corona chart, as they call it, which I think we relate to as something else completely these days, actually tells you a little bit about what's there. So what is really interesting is that in these, that about a third of the bacteria are unidentified. So we don't know who they are or what they do. However, the other 2/3, we know that cyanobacteria is about 15% of the bacterial population, or 13% overall. And 80% of those cyanobacteria are nitrogen-fixing cyanobacteria. Then you also have the other types of bacteria which all have special roles. So this was in the exclosure, so the ungrazed area. And now we'll go to the grazed site, and the difference is highlighted in blue there, where the rhizobial bacteria were present in the grazed site, but they weren't present in the ungrazed site. So that was interesting to compare. And also in the grazed site, there was a slightly higher proportion of cyanobacteria. 89% were N fixes, and in the rhizobia, there were N fixes as well. Interesting also in the grazed site, we had chordata, which was mice and snakes. So we picked up their DNA as well, so they were there too. So we obviously they're on the landscape there, but it just gives you an idea of how fine a picture we're getting with the DNA sequencing. So just in summary of those sequences, the bacteria formed 90% of the biocrust. Cyanobacteria, although they formed 15% of the actual biocrust bacteria, they're the greater portion in biomass, so that you can actually see them like in this picture here, with ants over them. So that's a really important part of the biocrust because it physically is the larger portion of the biomass. However, the other bacteria are all really important, and you've got N fixes, symbiotic bacteria, ones that work with decomposition, and also the cyanobacteria, which sequester carbon because they photosynthesize. And cyanobacteria are regarded as ecosystem engineers because they're ones that are able to modify the soil to suit and help plants as well. So the other part of our research is based at Wombiana near Charters Towers there, the grazing trials, and this is still part of this boosting the natural regeneration of the nitrogen capital in the grazing lands. Our research questions there are based around, are cows and crusts compatible, and how much biocrust is actually there, and are there biocrust hotspots and what are they? And we also want to know about the nitrogen, of course, because that relates to plant protein, which is always important. So we have done field data collections there across several sites. There's been heavy stocking sites, moderate stocking sites, and wet season spelling sites. And we've heard a bit about wet season spelling across these talks and how it works. So today I'm also going to give you the wet season spelling story from a biocrust point of view. We've also incorporated no stock controls. So we're looking at the biocrust community composition there with sequencing, as I've just shown you for Kidman, but we don't have those results yet. And the same with the N fixation tests, where we're using isotopic gases to trace, they're labelled, and we can actually see what comes from the air and goes into the biocrust because it's labelled. So those results are expected pretty soon. So the key results from Wombiana relate to the fact that in the red-yellow sands, which these, I'll just make a note here, these results were actually at a time when we'd gone through 2 fairly low rainfall years, so they're probably the sites at the worst stage before a good wet season last year. So the bare ground in this site was 2.5 times higher than everything else, it was about 80%, so that was quite significant, and the biocrust cover was significantly lower in the heavy stocking areas. In the duplex clays, what was really interesting was that the biocrust cover in the control and the wet season spelling were not significantly different. And so we were really interested in the fact that you had 34% cover there compared to 18% cover in just the moderate stocking. So the wet season spelling really did make a big difference to the biocrust cover. So from a biocrust point of view, why wet season spelling works is because you've got the cyanobacteria in the dry season, almost acts like a compost at the beginning of the wet season. So if you're actually giving it a time to recover, it regrows and recovers across the course of the wet season. So that little period of time to recover is all important. So spelling in the wet season is particularly good from that point of view. So one of the conservation of biocrusts is important in terms of the wet season spelling, cool fires and destocking during drought. Now, I'll just mention quickly monitoring bare ground because we have a lightning talk from one of our master students, and I encourage you all to look at it. And of course, bare ground has biocrusts on it too, so we're pretty keen to look at that area of research. So biocrust hotspots are important in the fact that they're a natural and obvious component of the landscape. They provide functional diversity and they're proportionate, and they are important because they're a natural asset. They also provide a micro-inoculant to facilitate regeneration post-fire, post-drought, and they generate biodiversity. So I'm hoping that you've all learned a bit more about biocrust today, and I've also got a live poll there which I'd like you to fill out. Thank you.